In this section, we will talk about the contractile mechanism in smooth muscles. First, we will discuss chemical basis for smooth muscle. Well, smooth muscle contains both actin and myosin filaments, having chemical characteristics similar to those of the actin and myosin filaments in skeletal muscle. It does not contain the troponin complex that is required for the control of skeletal muscle contraction, and thus the mechanism for controlling contraction is different. This topic is discussed in more detail later in this video. Chemical studies have shown that actin and myosin filaments derived from smooth muscle interact with each other in much the same way that they do in skeletal muscle. Furthermore, the contractile process is activated by calcium ions, and adenosine triphosphate, or ATP, is degraded to adenosine diphosphate, or ADP, to provide the energy for contraction. There are, however, major differences between the physical organization of smooth muscle and that of skeletal muscle, as well as differences in excitation-contraction coupling, control of the contractile process by calcium ions, duration of contraction, and the amount of energy required for contraction. Now let's discuss physical basis for smooth muscle contraction. Smooth muscle does not have the same striated arrangement of actin and myosin filaments as is found in skeletal muscle. Instead, electron micrographic techniques suggest the physical organization, which illustrates large numbers of actin filaments attached to dense bodies. Some of these bodies are attached to the cell membrane, and others are dispersed inside the cell. Some of the membrane-dense bodies of adjacent cells are bonded together by intercellular protein bridges. It is mainly through these bonds that the force of contraction is transmitted from one cell to the next. Interspersed among the actin filaments in the muscle fiber are myosin filaments. These filaments had a diameter more than twice that of the actin filaments. In electron micrographs, 5 to 10 times as many actin filaments as myosin filaments are usually found. To the right is a postulated structure of an individual contractile unit in a smooth muscle cell, showing large numbers of actin filaments radiating from two dense bodies. The ends of these filaments overlap a myosin filament located midway between the dense bodies. This contractile unit is similar to the contractile unit of skeletal muscle, but without the regularity of the skeletal muscle structure. In fact, the dense bodies of smooth muscles serve the same role as the Z-discs in skeletal muscle. Another difference is that most of the myosin filaments have side-polar cross bridges arranged, so that the bridges on one side hinge in one direction, and those on the other side hinge in the opposite direction. This configuration allows the myosin to pull an actin filament in one direction on one side, while simultaneously pulling another actin filament in the opposite direction on the other side. The value of this organization is that it allows smooth muscle cells to contract as much as 80% of their length, instead of being limited to less than 30%, as occurs in skeletal muscle. Now, let's discuss the comparison of smooth muscle contraction and skeletal muscle contraction. Although most skeletal muscles contract and relax rapidly, most smooth muscle contraction is prolonged tonic contraction, sometimes lasting hours or even days. Therefore, it is to be expected that both the physical and chemical characteristics of smooth muscle versus skeletal muscle contraction would differ. Now let's discuss some difference one by one. First is the slow cycling of the myosin cross bridges. The rapidity of cycling of the myosin cross bridges in smooth muscle, that is, their attachment to actin, then release from the actin, and reattachment for the next cycle, is much slower than in skeletal muscle. The frequency is as little as 1 by 10 to 1 by 300 that in skeletal muscle. Yet, the fraction of time that the cross bridges remain attached to the actin filaments, which is a major factor that determines the force of contraction, is believed to be greatly increased in smooth muscle. A possible reason for the slow cycling is that the cross-bridge heads have far less AT-pace activity than in skeletal muscle. Thus, degradation of the ATP that energizes the movements of the cross-bridge heads is greatly reduced, with corresponding slowing of the rate of cycling. Second is the low energy requirement to sustain smooth muscle contraction. Only 1/10 to 1/300 as much energy is required to sustain the same tension of contraction in smooth muscle as in skeletal muscle. This too is believed to result from the slow attachment and detachment cycling of the cross bridges, and because only one molecule of ATP is required for each cycle, regardless of its duration. This low energy utilization by smooth muscle is important to the overall energy economy of the body, because organs such as the intestines, urinary bladder, gallbladder, and other viscera often maintain tonic muscle contraction almost indefinitely. Third difference is the slowness of onset of contraction and relaxation of the total smooth muscle tissue. A typical smooth muscle tissue begins to contract 50 to 100 milliseconds after it is excited, reaches full contraction about 0.5 seconds later, and then declines in contractile force in another 1 to 2 seconds, giving a total contraction time of 1 to 3 seconds. This is about 30 times as long as a single contraction of an average skeletal muscle fiber. However, because there are so many types of smooth muscle, contraction of some types can be as short as 0.2 seconds or as long as 30 seconds. The slow onset of contraction of smooth muscle, as well as its prolonged contraction, is caused by the slowness of attachment and detachment of the cross bridges with the actin filaments. In addition, the initiation of contraction in response to calcium ions is much slower than in skeletal muscle, as will be discussed later. Moving towards fourth difference, and that is maximum force of contraction is often greater in smooth muscle than in skeletal muscle. Despite the relatively few myosin filaments in smooth muscle, and despite the slow cycling time of the cross bridges, the maximum force of contraction of smooth muscle is often greater than that of skeletal muscle, as much as 4 to 6 kg per centimeters square cross-sectional area for smooth muscle, in comparison with 3 to 4 kilograms for skeletal muscle. This great force of smooth muscle contraction results from the prolonged period of attachment of the myosin cross bridges to the actin filaments. Fifth difference is that latch mechanism facilitates prolonged holding of contractions of smooth muscle. Once smooth muscle has developed full contraction, the amount of continuing excitation can usually be reduced to far less than the initial level, even though the muscle maintains its full force of contraction. Furthermore, the energy consumed to maintain contraction is often minuscule, sometimes as little as 1/300 of the energy required for comparable sustained skeletal muscle contraction. This mechanism is called the latch mechanism. The importance of the latch mechanism is that it can maintain prolonged tonic contraction in smooth muscle for hours, with little use of energy. Little continued excitatory signal is required from nerve fibers or hormonal sources. Another difference is the stress relaxation of smooth muscle. Another important characteristic of smooth muscle, especially the visceral unitary type of smooth muscle of many hollow organs, is its ability to return to nearly its original force of contraction seconds or minutes after it has been elongated or shortened. For example, a sudden increase in fluid volume in the urinary bladder, thus stretching the smooth muscle in the bladder wall, causes an immediate large increase in pressure in the bladder. However, during about the next 15 to 60 seconds, despite continued stretch of the bladder wall, the pressure returns almost exactly back to the original level. Then, when the volume is increased by another step, the same effect occurs again. Conversely, when the volume is suddenly decreased, the pressure falls drastically at first, but then rises in another few seconds or minutes to or near the original level. These phenomena are called stress relaxation, and reverse stress relaxation. Their importance is that except for short periods, they allow a hollow organ to maintain about the same amount of pressure inside its lumen, despite sustained large changes in volume. This was all about this section. In our next section, we will talk about regulation of contraction by calcium ions.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.